Everyone. For those of you who I haven't had the pleasure of meeting yet, my name is Kyra Schaefer. I'm a second year Frontiers master's student in Marjorie Weber's lab here in EEB, and I have the pleasure of introducing our seminar speaker today. Dr. Wesley Bickford received his bachelor's degree from Indiana University in environmental science, his master's degree from the University of Maryland in wetland science, and finally his PhD here at the University of Michigan in EEB. Woo. Dr. Bickford has worked for the USGS Great Lakes Science Center within the Restoration and Conservation Sciences branch here in Ann Arbor for about 12 years. He started out as a research technician, but later returned to work as a pathways ecology intern while obtaining his PhD, and ultimately transitioned to a biologist position. His research focuses on wetland ecology, invasive species, and plant microbial interactions, with a particular interest in exploring restoration and invasive species management strategies rooted in molecular biology and plant-soil interactions. Wes is actually my former PI, and I had the opportunity to work with him after my undergrad on a multi-year research project that focused on invasive species management in wetlands. He has played an integral role in my personal and professional journey as a scientist, and I'm so excited for y'all to hear about his recent work. His talk today is titled Linking Mechanisms to Practice, Improving Outcomes in Great Lakes Wetland Restoration. Please join me in welcoming Dr. Wesley Bickford.
Well, thank you, Kyra. Can you all hear me okay? Is this good? All right. Well, thank you for the introduction. Yeah, it was a really nice bio. I wasn't sure if I needed to provide something, but it looks like she did her homework and looked all that up, so that's great. I'm really excited to be here today. As Kyra mentioned, I did my PhD here in this department and got to be one of the first students to move over to this building and take advantage of this really cool space. So it's cool to come back. And so I'm going to talk to you today about kind of what I've been working on kind of since I left at the work at USGS, and covering a little bit of the tail end of my dissertation research, so that you can see how it turned into some of the more applied work that we're doing at USGS. All right, so I always like to start a presentation with a really big picture of me. So there you go. Just a little bit about me again. I'm a biologist at the USGS Great Lakes Science Center, and I got this question a lot today about where it's located. We're right off of North Campus, so very close to here. And I'll keep coming back to that because I want to work with all of you if you're interested in working with us at the USGS. So off of North Campus, I'm part of the Great Lakes Science Center there. I'm also in a role as a visiting scientist with the Midwest Climate Adaptation Science Center, which is part of USGS. And my work at USGS is in the Restoration and Conservation Sciences branch. So that means that I think a lot about invasive species control. I think about wetland restoration. I think about how plant microbial interactions can improve invasive species control and wetland restoration. And I've been thinking a lot about climate adaptation and how to incorporate climate adaptation strategies into restoration projects. I'm gonna move over here so you can see me. No, I like this. Okay, I'm going to kind of ground us in where I work. So I do a lot of work in Great Lakes coastal wetlands. And although we're in the Great Lakes right now, I will venture to guess that many of you maybe have not been to coastal wetlands in the Great Lakes. You might have been to coastal wetlands on the East Coast or in one of the salty coasts and seen these really vast, wonderful ecosystems. And I will argue that we have very similar, vast, wonderful ecosystems in the Great Lakes as well. These really provide essential habitat for wildlife. They're really important for absorbing a lot of the nutrients that are coming off the landscapes that end up in the lakes and can cause harmful algal blooms, and for reducing coastal hazards that we even have in Great Lakes ecosystems. So they serve a lot of the same functions that they do in the salty coasts. They're really important ecosystems here as well. And there are lots of stressors that are impacting coastal wetlands in the Great Lakes. And I'm going to talk a lot about invasive species and some of the things that we're doing to control invasive species and restoration strategies. But this is an image showing where large Phragmites populations exist in western Lake Erie Basin. So all of that bright yellow is like a really dense monoculture of Phragmites. And so this just kind of goes to show you why this is an important issue for land managers in the region, to kind of try to control some of these infestations that are really taking over coastal wetlands and providing poor habitat. Additionally, climate change is a concern in coastal wetlands, as you might expect. And I have this hydrograph up here because it shows you the way that Great Lakes water levels fluctuate. And water level fluctuations in the Great Lakes have happened forever. I mean, water levels go up and they go down, and they're kind of driven by climatic patterns, the amount of snowfall, the amount of ice cover, precipitation patterns in the years prior. And so they do fluctuate, and they always have. But what we're seeing with increasing climate change is that these fluctuations are becoming more drastic. We're seeing, like in the past 10 years, we've seen an extreme high and an extreme low in the Great Lakes. And so the expectation is that these fluctuations may increase, making management much trickier. And so if you think about a corollary to like sea level rise in coastal areas, we at least kind of know the trajectory of that, right? So management, we know what to expect year after year, the water level going up. In coastal wetlands of the Great Lakes, it's difficult in the way that we don't actually know if it's going up or down predictably, and so we have to kind of plan for both and be prepared. So management is challenging. There are many efforts in the Great Lakes to restore coastal wetlands, and many partner groups that are thinking about these issues. And we work with a lot of them at USGS, and our work is really centered around the questions that partners ask, and we're driven by their needs. But it essentially comes down to a few things that they're all really interested in. They're striving for less invasives in coastal wetlands, more desirable native plants, habitat and water quality improvements, and even resilience to climate change, recognizing that these stressors are occurring across the basin. So our job at USGS is really to help problem solve for our partners. We're driven by questions that our partners ask. We develop research questions that can really respond to these needs of partners and help try to solve problems, do our best. So I'm going to kind of help you think about the way that I think about my research program, and title of my talk was Linking Mechanisms to Practice, and I want to kind of give you a sense of what I mean by that. If the goal is improving coastal wetland restoration outcomes, I'm doing that in my research program in a few ways. Thinking about uncovering ecosystem mechanisms, and what I mean by that is thinking about how species interact with one another and how that leads to changes that we see in ecosystems. Because understanding how those species interactions occur will help us improve interventions. And so it leads into improving these interventions that could be controlling invasive species, could be restoring native species in a wetland. And then I'm really also interested in this piece of evaluation. So understanding how do we ask the right questions about how well are we doing, and how do we monitor these sites so that monitoring data kind of feeds back into further improving those interventions that we're doing. Really central to all this is this co-production piece, and I mentioned this before. We're really responsive to the needs of our partners, and so it really it is pervasive in all the work that we're doing. Is that we're working with partner agencies and groups, and in some cases, on the research questions, here, well, let's study these things, and they're integrally involved in every aspect of the project, from designing the experiment to doing the data collection with us to helping report the results. So it is a really important aspect to all the work that we're doing. This piece here, this evaluation piece, I think is super interesting, but it's very new in my research program, and I'd love to talk to you about this if you're interested in this, but I'm not going to really cover it much in the talk today, so I just want to give you a heads up that that's not going to be something that's going to be covered. But I'm going to start by kind of giving an overview of some of these, a little bit about certain projects that have covered each of these aspects of my research program. So we'll start here, and this actually goes all the way back to what my dissertation work was on, and I'm not going to give my whole defense here, I'm going to try to keep it to like 3 minutes on this topic, but it gets to this idea of uncovering mechanisms and understanding what's driving invasiveness in plants, because that could help us better control these plants. So as a graduate student, I was really interested in what were the mechanisms underlying invasiveness. Is it things about plant traits that were really important? Is it these species interactions? And I was focused on studying species interactions and interested in mutualisms, antagonisms that might be driving how species were performing on a landscape. I got really interested in how plants and microbes interact with one another, knowing that there are microbes all over the place, right? There are microbes on the surfaces of leaves and in the roots and in the soils, and plants are interacting with microbes at all stages of their life cycle. So they're incredibly important to the plants. And so from, I came at these questions like very simply, like what are these things doing, and are they actually contributing to invasiveness in invasive plants? So I'm going to introduce you to an old friend of mine who you'll be hearing a lot about. This is the plant that I studied throughout my dissertation work and continue to study at USGS, and showed you the map of Phragmites populations across the western Lake Erie Basin. This is Phragmites australis, a very tall wetland grass that's found kind of all over the place in the Great Lakes region and throughout North America. It's really cosmopolitan invasive species, something that you will kind of see in any country in the world. And is it a particular problem in the Great Lakes because you see this incredibly tall biomass that can grow in these dense monocultures. And what you see above ground, you have to imagine there's as much, if not more, biomass below ground as well, because they have these really dense root and rhizome systems that form these networks that are, you can imagine if you're another plant trying to get up and have some space and live, it would be incredibly difficult. There's just like no room. Right, so it's, it creates these dense monocultures that are not really good habitat for other natives or for wildlife. And the reason that it's such a priority for USGS is it's not just an ecological or environmental issue, it's that it has societal impacts as well. It's really a nuisance across the landscape, as well as being an economic concern, as it can have property value kind of issues associated with it. There's also two lineages of Phragmites. So there's the invasive Phragmites, which I just shared a bunch about, and then there's a native Phragmites. And native Phragmites is found more in kind of high quality wetlands often, and it can be found in the Great Lakes coastal zone. We see it a lot in Great Lakes wetlands. So it's kind of co-occurs with others, with other desirable natives. It's in, it is not, as aggressive as the invasive counterpart. So my questions in my dissertation work were, thinking about the ways that plants interact with the mutualists and pathogens communities in the soils and in the roots. They're cultivating a community of microbes, and they're also kind of responding to that community of microbes. So there's kind of this effect that they're having and a response that's occurring within the plants as well to those microbial communities. And I wondered, do native and non-native lineages of Phragmites affect microbial communities differently? Are they cultivating different communities than one another? And do they respond differently to those microbes? So kind of like two different questions there. And that will help us kind of learn if those microbes are driving invasiveness in these plants. So this is me a few years ago when I had less gray on me, out in the field, pulling up Phragmites roots. We did a survey of the root endophytes in Phragmites populations and native and non-native populations of Phragmites, looking at sites where they co-occurred with one another. So these were kind of small stands of each, so we could find places where native and non-native both were occurring at the same site. We did that for root endophytes. We also did that for soil microbes. And then we did a manipulative plant soil feedback experiment, actually one of the first things that happened in the growth rooms downstairs, which was kind of cool. And I'm going to go through my results of my whole dissertation in one slide. It's pretty cool. All right. When we looked at the root endophytes, we found that there was not a whole lot of difference between the endophytes that cultivated the non-native versus those that cultivated the native. And again, these are kind of small stands. So what was colonizing those roots was about the same stuff at a particular site. When we looked at rhizosphere microbes, like this, it was a little bit more complicated in that we saw at these smaller stands, there was not a lot of difference, so fairly similar to what we saw in the roots. But when we had these larger monocultures, we did see some differences between the lineages. So this is the smaller kind of mixed sites. But when we see the larger monoculture stands, we saw some differences in the bacterial communities. So like they were starting to differentiate from one another kind of like over time as these stands matured. And then again, I said that we used this plant soil feedback experiment. This is down in the growth room in the basement here. To ask the question, do native and non-native Phragmites respond differently to soil microbes? And so we did a plant soil feedback experiment where we kind of like isolated different portions of the microbial community, looked at the responses. And one of the things that we found was like in doing a sterile versus live soil feedback experiment, I'm going to show you results that this is just an effect size graph. So anything above that 0 line means that it grew better in the live soil. Anything below means that it grew better in the sterile soil. And so asking this question about do they respond differently to microbes, what we're expecting if the native was getting an advantage from these microbial communities is that it would grow better in live soil. So like with the microbes, it would grow better. But we actually found that it grew worse. So it grew worse in the live soil. In the sterile, or in the native variety grew about the same in the sterile and the live soil. So it wasn't really impacted by the microbes. So we found they do respond differently, but not in the expected direction. So it's kind of interesting. So again, what we found was that because it was not responding in that expected direction, that they actually did worse around their microbes. We concluded that they weren't actually driving invasiveness. They're not the reason that these plants are invasive on these systems. But the fact that they accumulate negative microbes may be something that is important to invasiveness, maybe something that could enhance these invasive interactions. I'm going to describe what I mean by that in the next few slides here. So I got interested in this idea of like pathogen spillover. If you're building up a bunch of pathogens in your community, especially over time, if you're over many years, the microbial community is becoming more distinct towards these invasive variety, and maybe they're building up these negative microbes, that this could have implications for native species. And others have looked at this as well. This was a fairly recent paper from down in New Orleans that looked at communities of soil microbes around Phragmites and around native plants and found really high diversity and abundance of pathogens in the soil compared to in the native plants. And so they speculated that it was helping these invasive plants maintain dominance in these monocultures and potentially push forward into these into the native zones as well. Another really recent paper that came out that was on this idea of like over time enemies may accumulate in certain invasive plants that are generalists, and that may disproportionately impact the native plants around it rather than those invasive plants. So like these really tall, R-selected, fast-growing, big plants may accumulate these generalist enemies that are having more of an impact on the smaller native plants around them. So I like to think of this as like, if like a swarm of mosquitoes attacked me, I'd probably like run out of here, right? Like I'm heavily impacted by that. But if like that same swarm of mosquitoes attacked the Hulk, like it's going to be like, yeah, whatever. And so the Phragmites is the Hulk in this analogy, bad analogy. But it seems like that these large invasive, these large plants may not be as impacted by these generalist enemies that the native plants that are more K-selected may be a little bit more impacted by. So that has implications for spreading. Like if you have more of these generalist enemies in your soil community, you may be enhancing invasiveness in those plants. So it could be important for pathogen spillover, for allowing these invasive species to spread. But additionally, could be important, as we control these invasive species, how long do these pathogens remain in the soil, and are they impacting the native communities that we hope to come back? So if we are looking for regeneration from the seed bank, are these going to be impacted by these generalist enemies as well? So are there like some legacy impacts of Phragmites on the landscape that would be really important to understand for restoration purposes? So I'm going to talk a little bit about this idea of legacy effects, and I've looked at this in a few ways. And some of this is like still ongoing research, so there's not like data. But one of the things that we're trying to do is to understand what happens after Phragmites removal by looking at the microbial communities in Phragmites stands at like 80 different management units across the basin that are about to be controlling Phragmites. So we did these. We got community members to gather soil samples for us, which we are working on now, looking at the abundance of soil pathogens using qPCR to look at certain pathogen families in the soils. And then we're going to try to pair that with testing native germination in these soils as well to see if native germination is inhibited in some way. So that work is still ongoing. But we're started also thinking about these legacy impacts and how they, it's not only the microbial impacts, legacy impacts that could be important, but also thinking about like the amount of herbicide that we dump on these systems to control plants like Phragmites. So that's a legacy in itself too, right? Like, what about the legacy of these herbicides? Are there impacts to regeneration of native species and invasive species? And so I want to highlight a few student projects who have looked at this, because I think that these are interesting questions that will help us manage the species better. So I had a student, and maybe some of you know Nick, he's in the department now. I worked with Nick as an MRAD student, and he was interested in this idea of like, what are the impacts of herbicides on Phragmites growth, and we looked directly at seeds. This was actually a question that we got directly from a partner, like, okay, well, we spray Phragmites and it has seeds out on it. Are we killing those seeds too? And I was like, I don't think so, but we can look at that and we can see at what point it matters that there's herbicide in the system for those seeds. And so that's what we did here, where we had an experiment where we sprayed herbicide, glyphosate and imazapyr on seeds. We also did a treatment where we actually soaked them in these herbicides to see if we could get some sort of uptake. And then we looked at adding herbicide to the growth medium. So like, does it actually have to have some absorptive tissue, like it's actually germinated and starting to grow for that herbicide to have an impact? And somewhat unsurprisingly, that's what we found. That herbicide does decrease germination, but only upon uptake. So you can spray these seeds with herbicide, it doesn't matter. You can soak them in herbicide, it doesn't actually matter for their germination. But if you put them in a growth medium that has that herbicide and it starts taking it up, it's going to impact the plants. So you may think, okay, that's obvious, right? The plant has to take it up to do something. And it is, but also tells us that, like, okay, if there are residues of these herbicides in soils, that's when it becomes a problem, is if those residues remain. So I had another student interested in thinking about how long do those residues remain in soils. And so a student worked on trying to understand, okay, if we apply glyphosate and imazapyr to soils, and we then plant seeds. We planted seeds at 2-week intervals after herbicide application, and then harvest them after about 3 weeks. We were trying to understand how those herbicides were impacting germination of those seeds. And this was pretty interesting. So what we're looking at here, sorry if that's hard to see given the lighting, but what you're looking at here is this is the performance anomaly. So the difference from the control. So the control is that black line. So anything below that black line means it did worse than the control. And our red is the glyphosate, the blue is imazapyr. And so you can see over time, the glyphosate-treated pots started germinating just as well as the controls. But those imazapyr-treated pots were like dead. Nothing was growing out of those things. And it took like about 12 weeks or so before we reached a point where it's not different from the control in terms of like these germination rates were about the same. But those imazapyr treated pots were still inhibiting germination even after this graph shows. We kept these going for about 26 weeks, like 6 months, and they were still doing nothing. So glyphosate is microbially degraded and eventually it breaks down, and it seems like the plants are able to then respond after that herbicide breaks down. What I thought was really interesting about this is we also did this with Phragmites and found something similar where over time the glyphosate is wearing off and they're able to germinate. But what I thought was interesting about this is it happened faster in Phragmites. So a few weeks sooner, the Phragmites is able to take advantage of that soil and germinate and do just fine. So it seems like maybe there's potentially like a higher tolerance for these herbicides in that invasive plant than in the natives, which is something to consider, that maybe these invasive plants would be able to bounce back after herbicide applications better than the natives would be. And so there are several management implications to this that you have to think about the residues in native plant recovery. The imazapyr is especially potent, and so native plant recovery is going to be really difficult following imazapyr applications. And I think in general, what it tells me is that management strategies that limit herbicide applications may be more effective for native recovery in general. And I'm showing this paper here too because this came out like as we were doing this study. Someone who looked at this on a landscape scale and found very similar results that these herbicide treatments can reduce survival but not prevent establishment. And interestingly, they did not use imazapyr in their study. It was only glyphosate. So yeah, finally these glyphosate sites can rebound and plants can establish, but it might take a little longer. Okay, so I'm going to move on now to a space that's around this improving interventions category here and talk about another invasive plant control. And something that I just mentioned was like limiting the use of herbicides. And so one thing that we've done is to work on a study using the cut to drown technique. And so I'm going to describe kind of what we learned out of this experience using this particular intervention. So I mentioned a few control strategies for Phragmites, and you can spray herbicide, you can cut it, you can burn it, you can flood it, and you can do a variety of these things all at once to have some impact on success of the plant. But a strategy that's become increasingly popular, especially in Canada, is this cut to drown strategy, and this is because there's more restrictions on the types of herbicides that you can use over water in Canada. So they will cut the plants under the water to drown them. And so this is not a new technique that we developed, but it's something that there was not a lot of, there were not best management practices, for instance, out there on how to do this well. And so we undertook this study. And I'm going to do a little bit of I'm going to talk a little bit about the plant physiology so you understand how this strategy works. It's fairly simple, but it's helpful to understand kind of how this plant breathes to get a sense for why this would work and when it should work. So Phragmites takes advantage of convective through flow to get air into its roots and rhizomes to perform oxygen to perform essential functions within the plant. So air comes in the stomata. And goes all the way down into the rhizomes and is able to oxygenate the rhizosphere and then kind of breathes out through the old dead culms. So this is a common strategy for wetland plants, especially those growing in deeper water, to get air kind of going through the plants. But it's crucial for this management strategy because it can do this in low water, it can do it in high water, as long as a portion of the plant is remaining above the surface. It's like it's snorkeling. It's kind of just, still getting some air. Air can come in the green and out the brown, and it's happy. But if you cut that plant below the water line, you've cut off that flow of air into the plant, and so now it's going to drown. It's not going to be able to to survive for very long that way, unless it can regenerate a stem back up above the water, which becomes important. So managers were interested in this technique and interested in, what is the optimal timing for cutting? Like, when should you go out and do this if you're going to do it across the landscape? And also, are these rhizomes actually inviable after you drown them? Do you kill the plant if it is drowned for a certain period of time? What happens if a clone extends into low water? So, like, can a clone be rescued in its flooded area by its non-flooded area, for instance? So these were the types of questions that we were interested in answering. And so we got a grant through the Science Support Partnership with the Fish and Wildlife Service. We cooperatively worked on this project together. We had a greenhouse study and a multi-year field experiment. And the greenhouse study was mostly proof of concept. It was done here at the Botanical Gardens just to make sure that, yeah, we can kill these plants by drowning them. And we proved that in this study. So I'm going to talk about this multi-year field experiment, and I have to give Kyra a shout out here because she was integrally involved in all of this work, and also all of the great artwork is not me, that's her. Thank you, Kyra, for that. So we did this work in two spots. We worked in a coastal wetland and in an impounded wetland. The coastal wetland was in western Lake Erie. The impounded wetland was in near the Saginaw Bay. And we created some plots or used these patches of Phragmites that you kind of see there that were naturally forming to use this cutting strategy to understand if we could kill the plants. So they were subjected to treatments varying in cut timing and frequency. And we developed this strategy, or we developed our treatments based on the life cycle of Phragmites. And so I'm going to give you a little overview of the life cycle here. Phragmites is coming up from rhizomes like right now, like this time of year, putting up new shoots. And it's doing that by using the resources that are stored over the winter in those rhizomes. Those carbohydrates are turning into their new stems. They're putting up those shoots. They come above the water, photosynthesize throughout the growing season. They're producing or making new carbohydrates. At the end of the season, towards the end of the season, kind of at peak growth, they start translocating those new carbohydrates. Anything that's not being used to like create a leaf is getting put down into the rhizomes. So this is called the translocation period. And then they're storing those over winter to be able to put up a new shoot the next year. So this becomes important if you're thinking about like, when should you cut this plant? You might think that like an early cut is a really good time to do it because that's the time right after it's put up a new shoot, but before it's had a chance to like replenish any of those carbohydrates. Maybe you cut it all off, and that's the time to do it. Maybe it's better to do a late cut. So you do it after this full growing season, but before that translocation period occurs, before it's put a lot of resources back into the rhizomes, you cut them off there. Or maybe you're crazy like we were, and you decide you're going to do it every 2 weeks and go out and just like not allow it to put any more carbohydrates into the rhizomes and make it deplete what's already there. So these were the three treatments that we applied in the field. And we measured success in these plots by looking at above ground growth, just at stem density that came back after our treatments. We looked at the amount of carbohydrates in the rhizomes. So I'm thinking of that as like the growth capital. Like, what does it have left in the tank to be able to put up a new shoot? And then viability. So we cut pieces of rhizome and took them into the growth chamber and allowed them to put up a new shoot to see if this is actually alive or not. Okay, so I'm going to share after the first year of cutting here. And what you're looking at here is, again, kind of like a deviation from control here. So these are our treatments and how they fared against our control. So if they're below that zero line, they did worse than the control. And you're looking at a 95% confidence interval around those points. So we found that we had significant reductions in all of these growth metrics after the first year, save for that early cut in rhizome viability. We found that continuous cuts were more effective, but crucially, while we were reducing the number of new shoots that were coming up out of those rhizomes, when we looked at viability, they were still alive. So they were actually still alive. So it didn't really matter that they were producing fewer shoots. It's like, well, they're still alive. They're going to be there the next year. And it told us that this is a multi-year process, not something that we can expect to accomplish in 1 year of cutting. So after 2 years of cutting, though, we found that the above ground growth capital viability, all of those were even more decreased. And pay particular interest to the carbohydrates in these treated plants. So they're really, really decreased to the point where in some of them there's just kind of like nothing left in the tank, which led to all of our continuously cut plots, the plots that we cut every 2 weeks, just completely died. Like nothing came back after 2 years of continuous cutting. So we were able to control these plants by continuously removing the biomass and depleting those rhizomes over the course of that experiment. And this was actually just published like 2 weeks ago in the Journal of Great Lakes Research, so you can read all about all the nuances that I didn't get to cover here. All right, so there was another really interesting question that I wanted to cover a little bit, and I had another student who was interested in this, and that was just what are these adventitious roots doing on these plants, and are they important to the success of this management strategy? And what I mean by the adventitious roots are these that are growing above the water, or above the soil but below the water, so like these water roots. And so I had a student who was an intern at the time who decided to work on this with me for her master's in Cs, and developed a cool experiment to understand what are these adventitious roots actually doing. It's been found in other studies, in other aquatic plants, that they can provide dissolved oxygen to plants in submerged water. So if they're doing that, that could be really important. Because, I mentioned like these plants have snorkels, and if you remove their snorkels, they die. But if you also are giving them an oxygen tank, right, like that's, they're going to live, right? So it's going to reduce the ability to kill these plants if they have these adventitious roots. So in her experiment was pretty cool in that she set this up at the botanical gardens, where she grew Phragmites and forced them to create these adventitious roots by growing them in high water, which all of them did after a period of time. And then did a couple of different types of cuts, so like a no-cut control, but then a partial cut and a full cut. And what's important about the partial cut is that in the partial cuts, these adventitious roots remained there. So there's like a little bit of stubble, and there's still some roots remaining, so it looks like this. And the really interesting thing I thought about all of this is that, wow, when you do a partial cut, the plants are putting up so many more new shoots. They're just like exploding with significantly more new shoots than if you do a full cut or no cut at all. So they're just like responding by putting up new shoots. And the question is then, is it because they have these adventitious roots attached to them? And so it seemed like the cut plants with these adventitious roots produced a lot more new shoots. And this is something that we could kind of see in the field, that like in certain sites, especially those that had the high prevalence of these roots, we saw like this amount of growth within 2 weeks when we came back. We're like, oh man, we just got all these down. How is this still alive? And here's a video of me cutting these. And I have like this, it's like a hedge trimmer that can go underwater, and I'm trying to get these things cut. And so you can see how it's like kind of hard, right? To get all the way down to the soil surface. And so what you end up with a lot of times is like the stubble on the surface that has these roots attached. And so I think that this is important, because if you're not getting all the way to the soil surface, you may be allowing these plants to have kind of defense against this kind of strategy. So I think actionable outcomes for managers is that cut to drown strategy can be really effective at controlling Phragmites, but you also need to think of it as like starving the plant of resources. It's not just about getting it underwater, but actually making it deplete those resources so it's not going to be able to come back up. We also found that it's important to cut near the sediment surface through previous work that it suggests that if you cut low, it may be better than just cutting anywhere under the surface of the water. And then importantly, like this is a super labor-intensive process, and I'm sorry if you can't see the lighting there, but like literal boatloads of frag that we were pulling in from out in these plots. So it was really labor-intensive, something that managers need to consider if this is something that they want to do. And but crucially, in year 2, these plants were producing a lot less biomass, and it was a lot easier to accomplish their treatments then. All right, so I'm going to move on. A lot of people today were asking me, like, what's your favorite thing about the work that you're doing now? And I was like, yeah, I enjoy the invasive species work, but it's really nice now to work on restoration projects where I get to be in like nice wetlands instead of all the worst wetlands, right, where there's tons of Phragmites everywhere. So I'm going to talk now about some of the things that I'm really excited about, and that's work looking at the impact of microbes on native restoration. And this, again, kind of falls in that improving interventions category. So I've been really lucky in that I've recently formed a partnership with the Bay Mills Indian community in northern Michigan in the UP to explore the impacts of or how microbes interact with wild rice. Wild rice, or manumin or monomin, is a culturally important plant to indigenous communities across the Great Lakes region. And so they're all really interested in improving restoration strategies around wild rice and getting more wild rice and wetlands across their territories and across the basin in general. And so I'm going to tell you just a little bit about the life cycle really quickly. This is an aquatic plant, grows in wetlands and deeper water in really high water quality environments. These seeds, this is a grain that germinates underwater. It has to stay hydrated. So it germinates underwater, and it lives part of its life cycle as a submerged plant. And then eventually that plant grows to the surface, and it has like a floating leaf stage where the leaf floats on the surface. When it gets enough root support, it eventually will have aerial shoots, and so it'll grow and be an emergent plant. It will flower and then produce seeds, and those seeds eventually then fall back into the water. All right, so I'm going to focus mostly on these two parts of the growth stage, so kind of the seed ripening and dormancy and germination phase for the next several slides here. All right, so, okay, I'll try to go faster here. I think of this in a very similar way to like the invasive plant research, where I'm trying to understand how these seeds and these plants are interacting with microbes, and how they are, those microbial communities are changing from site to site, and how site conditions impact the microbial communities that are around these plants, and how those impact the restoration of the species. So you can think about it in a very similar way, that they're interacting with mutualists and pathogens in those soil environments, and that those could be impacting the success of those plants on the landscape. So one of the things that we asked is, like, does it matter where you store seeds? So like one of the things to know about restoration process is that since these seeds need to stay hydrated, often tribes will like get seeds from their source population, transport it to a site that they're going to restore, and put it on like immediately. That's one way to do it. Another way to do it is to store those seeds over winter in a location like in a grain bag in a wetland area, just in the water over winter, and then spread them in the spring. And so I think about these as like all different ways or different pathways for inoculation of microbes. They could be getting microbes from their source population. They could be getting microbes from their restored site. They could be getting microbes, gaining microbes from this intermediate site that they're overwintered at. So we were asking, does it matter where the seeds are stored over winter, and can the microbial histories impact germination rates of these seeds? And then also, like, how do local conditions impact germination? So I had a student first ask, we were asking the question, do legacy microbes impact germination? So do the microbes from their source population impact the germination rates of these seeds? So we sterilized some seeds after we got them from a source population. We did this in two ways, one with a hydrogen peroxide, it was kind of a weak hydrogen peroxide, and another where we just tried to wash them off without using any acid, and we germinated them in the growth chamber. And we found that the sterilized seeds actually did germinate a little better, just kind of modest increases, but significant. And these washed seeds didn't really, the washing didn't really have an impact in terms of being significantly more of them germinating than the controls. We also looked at the microbial communities on these seeds, and we found that we did a pretty good job of reducing diversity by sterilizing these things. So looking at bacterial diversity here, and we saw that was drastically reduced in these sterile in our sterile treatments. But the washed was about as had just about as high a diversity as the control. So maybe the washing wasn't doing enough to remove those microbes. So this tells us that legacy microbes may be important in inhibiting growth, but also there's a little bit more to understand about what is it about the treatment to remove those microbes that may have actually inhibited or increased the germination rates as well. So more that we're looking into. We also were interested then again, how do the site conditions impact germination? And so I had a student looking at seeds and soils from different source populations, and they were overwintered, or from the same source population, that were overwintered in soil and water from different sites. And we had sites that had healthy populations of wild rice, some that had restored populations, so more sparse populations of wild rice, a naive site that just didn't have wild rice but looked like it could support it, and then our control. And you can kind of think of these as different densities, that we had high density, low density, and not present. And what we found when we looked at germination rates was that the highest germination was in our sterile environment, and the lowest was in the high density site. And if you look at this, you start to see, well, is this negative density dependence here? Are we starting to see some signs that maybe there's a feedback that we can detect if we look into this? And of course, is it microbial, or are there other reasons for this? And what about nutrients? All things that we started looking into in these little containers. And so we did some sequencing, and we looked at the microbial communities. And if we look at the soil bacterial communities, we did see really strong differences in the communities of bacteria present in these tubs that had different source populations, or different soil from different locations. And so the healthy site soils were very different, had different microbial community than the naive and the restored sites. And so, we saw differences in germination. There's now this correlation with differences in the communities of bacteria present. But we want to know a little bit about function. Were there some functional reasons that this could be occurring? So we've used these databases that can tell you a little bit about function, and this one called FAPROTAX that is supposed to kind of tell you what functions are potentially involved in the communities of microbes that you have. And we found some, and what I'm showing here are only those that were significantly different among our treatment groups. And so we had things like, chloroplasts were more likely in those healthy sites, and maybe that's a nutrient thing that's driving that. We also saw that these intracellular parasites were more prevalent in the healthy sites. So these are things this is very fresh data, but it got me thinking is this evidence or some evidence of a microbial feedback where some negative feedback may be occurring, or there's more pathogens or parasites in these healthy sites? Looked at the same thing for soil fungi, and similarly, we had differences among our treatment groups in their communities of fungi. And we could see some differences in the types of trophic modes that we're finding, although it didn't fit this pattern very nicely. The only thing that we could say is that, there tended to be a little bit more pathogens in the healthy sites than in these naive or restored. But it was also pretty similar to the control. So it's kind of what I'm getting at is we're up against there's a barrier there in terms of what we can know from doing DNA-based analysis in these environments. So what we need to do is to look for evidence in the field, which we're doing now, or we're trying to look for community differences at different field sites and correlate this with stem densities that we're seeing in the field, success of restoration in the field. And we've been working on some of this over the past several years. And then one last thing that I've been really excited about is we are going to be partnering with a group out of Loyola University, a group that many of you might know from working at the biostation and the Team Typha. They've been working on developing biochar from the Typha that they've been harvesting, and finding that it has been improving success of restoration of some native plants, including wild rice. And so we got in touch with them because they're interested in understanding the microbial implications of applying this biochar. And to me, I was like, I'm super excited about this because we've been looking at these trends across the basin and things like that, but we haven't had a good intervention to where we can kind of see what might be driving changes. So now we have this biochar that we can apply. We can see changes in the wild rice productivity, and then we can also see if there are changes in the microbial community that could help us kind of understand what's happening. So I'm really excited about this collaboration, and we have a couple of tribes involved in the collaboration as well. Okay, so all this work has been really it's been really crucial to have this input from the tribal organizations. The tribes have been integrally involved in collecting the data with us and developing the questions and obtaining funding with us. And it's just a really great experience to be working with those communities. As I mentioned before, I'm not going to be able to talk about this last evaluation piece. And I'll just kind of sum up what the work that we've been doing. Is that our lab is really focused on these actionable, partner-driven research questions. And what I think has kind of defined my research lately is to be able to use microbial ecology as like a lever for restoration. Like, can we understand what do we know about how these systems interact with microbes, and are there tweaks that we can make that will improve the strategies that we're already using or develop new strategies? So I'm really excited about the future of that work in these restoration projects as well. I would like to say thank you to all the partners that have worked on this work, and as I've mentioned many times, they're really necessary for helping us develop the questions and have played a strong role in pushing the research forward as well. So I'd like to say thank you to all of them. Also, thanks to all of our students and interns and technicians who have done a lot of this work. And I was careful to kind of highlight students who have worked on this work, because I also now want to say, faculty members, if any of your students are interested in doing more applied work or taking some of the concepts that you all are working on, putting them in an applied setting in the Great Lakes, I love working with students, and it's not just me in our office. We have a full research staff that would love to have more student involvement. And one of the reasons I said, we're just down the road, you should call us up, we'll work together. I would love to do that, for real. So thanks to everyone who's been involved. And thank you all for being here. And now I can have a couple of questions.
I think we have time for a quick question or two. Does anyone have any questions?
Yes. Hi. You too.
It's nice to see those students too, because two of them are in classes I'm teaching right
yeah?
now, actually.
Cool.
Yeah. A lot of your research is focused on work in the wetlands themselves and working with the invasive species. The cause of these problems is humans. Mm-hmm. Is the USGS developing strategies to manage us?
That's a really good question. Yeah, so we're involved in some of these water quality agreements, things that are thinking about the sources of some of the nutrient pollution, that sort of thing that's occurring. I think that's the way that I can answer is that we're thinking about ways to work with other partner organizations like USDA or things like that we can help, stop these problems before they become, major issues. I think the answer is probably less than you would like to hear in terms of how much they're doing to manage people. Yeah.
Okay, cool stuff. You mentioned that the cutting and drowning seems like a really promising strategy, and it's cool. But you also said that it's pretty labor intensive.
Yeah.
But you quickly showed a picture of some sort of nifty gadget they seem to be using in Canada to mow it down. What is that machine, and is it have potential here?
Because it has the coolest name in the world. It's a truck saw. So it's called a truck saw. It's like this crane cutter that you can put on like a barge and cut things down underwater. And I think then you have a mechanism to kind of lift it on. So yeah, if you have funding, if you know you're going to be doing this for a while, you can invest in something like that. And obviously it's going to be a lot easier than what we were doing, which was like this, and then grabbing. And Kyra knows that this was hard, right? Really, it's not something that you could do on a large scale using the tools that we had. But there are tools out there that are better at that. So yes, I should have mentioned that in that slide, but I'm glad you picked up on that exists. Yeah. Oh, sorry. Here. Chris.
Hi, Wes. Nice to see you.
You too.
I was just wondering about the invasiveness of the European Phragmites, and in restoration projects that are focused on old field restoration, typically the native plants do best on the least fertile soils. And then if you fertilize them, that's when all the invasives come in and just take over, and it becomes very difficult to manage and make space for the natives. So in this case, is the Phragmites taking over in part because of nutrient pollution and sort of all the changes that have happened in wetlands? And then given that, is that nutrient addition from fertilizers going to make it very difficult to just bring back the native species?
It's a great question, and I think as for the reason why Phragmites has become such an issue, I think that is a huge component of it, is the nutrient pollution, and you see that when you look at where it's the most